You want to find your talk?
There we go.
Great. Okay, so we're pretty much on track. Jenny's going to talk to us about UV photodissociation to more completely characterize intact proteins, including post-translational modifications.
Thanks. Okay, thanks. Well, first of all, it's a great honor to be here today to celebrate with Ben and his award. So I'm happy to be here. So today I'm going to talk a little bit about some of our work over the last few years aimed at developing new ion activation methods to characterize large molecules. And so the technique that we've adopted is photodissociation, and that's using photons to energize molecules and do fragmentation. And so the overarching question that we've been trying to address with development of this technology is, can we improve the determination of sequences and modifications of complex mixtures of molecules? And you can imagine how important this is, whether looking at nucleic acids and how they are modified, for example, interacting with cancer drugs, or the complex structures of bacterial lipopolysaccharides that decorate the surfaces of bacteria and determine how pathogenetic they are. But today I'm going to focus on the realm of proteins and how post-translational modifications can be identified. And in order to do this, really what we need are more informative molecular fingerprints. And in the mass spec world, we think of that in terms of fragmentation patterns and interpreting those fragmentation patterns to reveal more information. I'm going to talk about 3 different strategies that we can apply with photodissociation to look at features of proteomics. So I'll run the gamut from top-down methods. That's where we're looking at intact proteins. And if we're looking at complex mixtures, in many cases those are more simple mixtures because we have not digested the proteins into smaller peptides, all the way to bottom-up methods, which is the traditional method, where you do use proteases to create small, very manageable peptides that chromatograph and ionize well, but they create increasingly complex mixtures when you're starting with large mixtures of proteins. And then in the middle down methods, where you use limited proteolysis to create mid-size molecules, very, very large peptides. So the mixtures are not going to be as complex as the bottom-up methods, and you retain more of the integrity of these larger size peptides. And so if you look at the whole workflow for proteomics in general, this has been adapted for decades now, starting with cells, extracting the proteins, then typically in the bottom-up methods using some sort of protease to create small peptides, which are then analyzed by some LC-MS method. Or if we look at the top-down methods, there we simply skip the proteolysis step, look at the intact proteins, and try to fragment those, interpret those directly. Regardless of these methods, they call out for very fast and efficient, robust ion activation methods to generate interpretable fragmentation patterns that can be then used with database search and scoring algorithms and de novo methods, in some cases, to really sort out the information and piece it back together in a constructive manner. And so if you're thinking about developing a fragmentation method, you're focusing then on activation of these ions to get them to cleave into meaningful ways. And so the attributes you'd like to promote with an activation method is you'd like something to be fast so that it can be used in a high throughput method. You'd presumably like high energy deposition to create a large array of fragments, especially for large molecules. Perhaps a large diversity of fragment ions to increase confidence in assigning structures. You'd like it to be very efficient, especially when dealing with low abundances of peptides or proteins, and so forth. And including, you also might like to dream big and make it effective for both positive and negative ions to really squeak out more information from the proteome. And we think all of these can be achieved using photodissociation, in particular UV photodissociation using 193 nanometer photons. And so in the simplest case, one can achieve photodissociation in a mass spectrometer by interfacing a laser to the mass spectrometer. And I show here, this is one of our earlier methods using a simple ion trap system. And so the important aspect of this is that all these, the information that I'll show today was acquired using commercial ion traps, which we have simply modified in a relatively simple manner by adding a laser. And so ion traps are a great fit for this because then we can store the ions and irradiate them with photons. The key is that we just need a way to get the photons to the ions. And so, for example, with this system, it's a linear ion trap, and we can bring the laser in from the back end. We drill a hole in the back end of the mass spectrometer, put on a flange with an optical window, and then we're ready to go. And so in this case, we're bringing the laser in. It's a UV laser that produces 6.4 EV photons. So each photon deposits quite a bit of energy, and it can intersect the ions when they're stored in the linear ion trap. Now I show one example up front so you know right away that there's quite a big difference between activating ions by collisions, which is the common gold standard method, and using UV photons. And so for this peptide shown here with the sequence given alphabetically, the top spectrum is the collision-activated dissociation spectrum. This very nice sequence coverage for this peptide, but if you look closely at all the ions, they're labeled with B and Y labels, which is very common for collision-activated dissociation. It works very well for peptides. The bottom one is a more congested spectrum because with UV photodissociation, in this case using the 193 nanometer photons, with a single 5 nanosecond pulse, we're generating AX, BY, and CZ ions. So clearly the mechanism for activation and fragmentation is quite different, and we've got a larger diversity of ions. And so what causes that is really, if we think about the processes of collisional activation versus photodissociation in an ion trap, collision activation I've illustrated here, where we have some sort of precursor ion, stable ion in an energy well, and the collision-induced dissociation in the trap involves a multi-collision stepwise process in which the energy is deposited in vibrational modes, ultimately heating up the ion sufficiently so that we get cleavage of the most labile bonds and fragmentation. This is what leads to the BY ions. With the UV photodissociation, this is in many cases just a single photon event, a single step of energy, 6.4 EV, so it's significantly higher energy deposition. That can also not only access excited electronic states, but there also can be internal conversion and then vibrational energy redistribution, leading to this very diverse arrangement of fragment ions, including A, B, C, and X, Y, Z ions, cleavages at every point along the peptide backbone. So now I'm going to show how this UV photodissociation method can be used in the various opportunities for proteomics, starting with, let's say, a bottom-up strategy. And this one I'm going to mention briefly, involving analysis of the acidic proteome, focusing on negatively charged ions. This is really a little bit of a shortfall of collision-induced dissociation methods, which work wonderfully for the more basic side of the proteome for positively charged ions. But in fact, there are proteins that are decorated with acidic modifications, such as phosphate groups, sulfonate groups, sialic acids, or stretches of proteins that have acidic residues, such as aspartic and glutamic acids, which may not ionize so well in the positive mode, and having opportunity to look at the negative mode could be a good fit. And so as we said, we know that collision dissociation works great for positively charged peptides, as shown here for this peptide. It's got basic sites, ionizes well in the positive mode, and we see a nice array of B, Y ions that allow us to cleave almost every backbone position and give very good sequence coverage. For a comparably sized peptide, this one is a more acidic peptide. We've ionized it in the negative mode, and this is where the collision-induced dissociation pattern seems to fall a bit short. Mainly the only fragment ions we see are loss of water molecules, which is common in the negative mode, but it's uninformative. We really don't see many sequence ions. so giving low sequence coverage. And so now we have a comparison of CID on the top to UVPD for the same peptide. This is a phosphopeptide human aquaporin, doubly charged in the negative mode. CID pattern, again, we see mainly loss of small neutral losses like CO2, water, or phosphoric acid. We see just a couple of cleavages, so the sequence coverage is not particularly comprehensive. In the bottom case, with UVPD, we mainly generate AX ions in the negative mode, and we see more extensive sequence coverage, which allows us then to use this negative mode to look at the acidic proteome. As one example, one of our collaborators was interested in protein kinases, in particular mitogen-activated protein kinases, which have large acidic stretches and thus would anticipate be well ionized in the negative mode. And here's an example where this is a nano-LC run, as one would expect for a typical bottom-up experiment. And I've just highlighted a couple of the regions where peptides eluted, and you can see these nice fragmentation patterns in the negative mode for a -2 and a -3 charged species for a couple of these peptides from these protein kinases. Now, a really challenging application is when you're looking at specifically at a substance that generates a very acidic proteome. In this case, for example, an archaic gram-negative microorganism called Halobacterium solanarum. It's considered a extremophile, which thrives in high salt environments, and it's actually a good model system for biologists and biochemists who are trying to study how organisms adapt and survive in very extreme conditions. It's got this very characteristic red color due to carotenoids in the organism. But in fact, its proteome is very acidic. And so just looking at some results for UV photodissociation versus conventional collision-induced dissociation, negative mode for UVPD, positive mode for CID, here's an example of one of the nice fragmentation patterns for one of the peptides from that halobacterium in the negative mode. And we'll go straight down to the summary here. The total number of proteins and unique proteins for the UVPD in the negative mode versus CID in the positive mode. Once again, CID in the positive modes performs admirably. You have nearly 1,000 proteins identified. In terms of unique proteins relative to UVPD, there were 246. And when we look now in the negative mode with the UVPD results, 800 proteins identified, and in this case, just over 80 unique proteins that were identified that were not found by CID. So if some of those were proteins of particular interest, they might not have been seen by CID alone. And so the combination of the positive and negative modes really adds a more comprehensive look at the proteome. And here we did the same comparison for that halobacterium using UVPD in the negative mode versus ETD, CID, and HCD. In each case, the number of proteins identified is illustrated here. And from this Venn diagram, you can see which proteins were found by one or two or all three methods and the number of unique proteins for each one. And you perhaps don't want to necessarily spend the time combining all four methods, but it seems that strategic use of a negative mode method plus one of the positive mode methods would give you the largest proteoform characterization. And so we can see if we look at this using a plot of isoelectric points versus the number of proteins identified, we can see that there is a more of the more acidic proteins with lower PI values found for the UV photodissociation, which is the red trace, relative to the other three, which looked at the positive mode and therefore identified proteins and peptides that were more ionized more favorably in the positive mode. Okay, so that was a good example of a bottom-up method using the negative mode for photodissociation. And that, in fact, is the most common method now for proteomics, using the bottom-up method where you are, in fact, digesting proteins into peptides prior to analysis. The opposite extreme is to use the top-down analysis, where you, in fact, look at the intact proteins. These can still be complex mixtures, but they have not been subdivided into peptides. So with chromatographic separation, you still need ability to identify individual proteins through the fragmentation patterns. And so this, in fact, we've implemented on a higher performance mass spectrometer like an Orbitrap mass spectrometer. That's because if you're dealing with larger proteins that will create multi-charge fragment ions, you really need that higher resolution and higher accuracy to assign those multi-charge fragment ions. And so in this case, we're doing photodissociation in the HCD cell of the instrument. So again, optical window at the back end, a couple of mirrors, and we've got our laser in the HCD cell. In this case, then the ions after fragmentation will be sent to the Orbitrap for high accuracy analysis. And so just an example, here's a model protein myoglobin, 153 residues, about 17,000 kilodaltons. There's the electrospray mass spectrum. We would isolate one of those charge states, for example, the 20 plus charge state, and irradiate it with 193 nanometer photons. That gives a fragmentation pattern that looks like this. This, for example, was acquired using a single laser pulse at 2 millijoules. It looks fairly congested to the naked eye. If you blow up those regions, you can see that they actually entail isotopically resolved fragment ions that can be assigned based on charge state and mass. And so it's actually an interpretable pattern, even if it looks congested at first glance. And if you look closely at those fragment ions, you see there's, in fact, a large array of different types of fragment ions, just as we saw before with the small peptides. With this technique for proteins, you see A, B, C, and X, Y, Z ions. All of those can be formed with this high-energy photons. For those less familiar with this technique, it's always nice to compare how this performs relative to other MSMS methods. So here I've shown a bar graph showing different charge states of that same protein, myoglobin, and the sequence coverage, basically how much of the backbone we're mapping with this technique for 4 MSMS methods. ETD is the blue bars, CID is red bars, HCD is green bars, and then UVPD are the purple bars. And basically, one would prefer a higher level of sequence coverage. And you can see for ETD, it actually gets a little better with a charge state averaging up to about 50% sequence coveraging. This collision-based methods, which are CID and the higher energy HCD methods, their bars tend to go down with higher charge states, which is common for these proteins and collision-based methods. And UVPD actually outperforms these for this protein myoglobin with close to 90% sequence coverage for quite an array of different charge states. Here's a slightly step up to carbonic anhydrase, which has 259 residues and about 29 kilodaltons. And this fragmentation pattern here can be mapped based on the entire alphabetical structure of or sequence of the protein, with all the cleavage marks coded to show whether you're forming A, B, C, or XYZ ions, or combinations of both. And it ultimately resulted in 87% sequence coverage. If you look at this little map here, you can see that the A ions were in fact the dominant type of ions formed, with lesser amounts of BC ions. And from the C-terminus side, you can see X, Y, and Z ions are all quite predominant. When we compared HCD, the higher energy collision-based technique, for the same charge state, we attained about 30% sequence coverage. And the reason one does see higher sequence coverage for UVPD is that these fragmentations extend deeper into the sequence of the protein. And so this histogram shown here compares CID, the higher energy HCD, and UVPD for 5 different protein variants of the green fluorescent protein called G3, G2, the wild type G12, and G13. So it's just 5 different proteins. And what we're showing is the number of diagnostic fragment ions ranging from the first 80 residues of the protein to the middle 80 residues of the protein to the terminal 80 residues of the protein, going from N terminus to C terminus. And these are just all the N-terminus ions. You can see the dominant number are formed in between 1 to 80 residues. So those are fragments containing 1 to 80 residues. And UVPD has the greatest number of those diagnostic fragment ions. As you go to the middle section of the protein, UVPD still performs quite well for creating quite large fragment ions that can be identified. The others have really dropped off in their ability to form those fragment ions. And then as you go out to the final portion of the protein, UVPD is still generating quite large fragment ions that aren't really observed to any great abundance for the other techniques. One always asks if this then technique can be adopted to LCMS workflow for higher throughput. So here's which is one example of a chromatogram obtained for a gel-free fraction of a yeast lysate adapted for LC separations of proteins using a C4 column. And this is just one example, one protein selected here, eluting about time point 50 minutes. This was identified as a eukaryotic transition initiation factor 5A. This is its sequence. You can see all the cleavages shown here give good coverage of this protein. It's about a 17,000 kilodalton protein. And also some modifications are identified on this protein, N-terminal acetylation and unusual lysine modification. This data was acquired using a single pulse per LC scan. Okay, so in addition, we did a comprehensive analysis of some more complex mixtures of proteins. These are ribosomal proteins. We pulled out about 25 of the ribosomal proteins that were identified both by UVPD and the higher energy collision activation technique. And this was also done in LC mode. And what I've shown here are these different proteins just labeled by their common ribosomal names or numbers. And here's the number of identified fragments that help to characterize those proteins. And you can see for HCD, the average number of fragments falls in the 50 to 100 range. For UVPD, it's 100 to 200 fragment ions that can help identify those proteins with higher confidence. So across the board, UVPD almost always outperforms HCD, except for a few up here, which we're still exploring. And so then the final few minutes of the talk, I'd like to now look at the intermediate option for proteomics. We've looked at the bottom-up method where we digested the proteins into small peptides, the top-down method, which is challenging because you are working with intact proteins, and that's a real burden for the chromatographic methods. And now let's think about the compromise, the middle-down analysis. This is where we do a partial digestion or a limited digestion to create, instead of small peptides, very large peptides, ones that are 3 kilodaltons or greater. This, in fact, creates mixtures of moderate complexity because we don't have a lot of small peptides, but it's certainly more complex than the intact proteins. However, we can use chromatographic methods that are more similar to peptide-like analysis, so that's a bonus for these middle-down methods. And the sheer size of the molecules makes them more adaptable for database searching methods. And so I'm going to illustrate this type of middle-down analysis for characterization of antibodies. Now, antibodies that most of you are familiar with are simply large glycoproteins, and these would be a real challenge for top-down methods because they are on the size of 150,000 Daltons. So even for UVPD methods to obtain a fragmentation pattern, which is feasible for using UV photons, the fact of the matter is we don't have sufficient resolution with the mass spectrometer to sort out the very complex fragmentation patterns. Just too many overlapping charge states of fragment ions, too many overlapping isotopic distributions of fragment ions, so it's really quite a mess if we go to proteins that are 150,000 kilodaltons. So that means a middle-down approach looks very attractive. Now, if you think about this antibody, it consists of 2 identical heavy chains, which are the cartoon-like structures in the middle, and then two shorter identical light chains, which are the ones shown on the outside. These are all bonded together using with disulfide bonds. So that's what creates this large glycoprotein. The key part about this is that many antibodies have very homologous sequences. So there's large regions of the sequence from different antibodies that are identical. So if you do chop these into very small peptides, you will get large amounts of peptides that are identical from antibody to antibody, and thus aren't helpful in identifying unique antibodies. It's actually, in fact, these complementarity determining regions close to the ends here on, for example, these light chains, which are really the more unique sections, and they also are the sections that mediate antigen specificity. So it's real important that we identify these sections above all. And so if you think about using a middle-down approach, you want to use a protease which will cause larger peptides. And so here we're using something called lyc, which cleaves after lysines. It should generate peptides in the 1.5 to 10 kilodalton region. And as shown here, if you look at an in silico map of what you would anticipate for digestion using this lyc, each of the peptides ends with a lysine, and they're fairly substantial size peptides. In addition, the blue regions are some of these key complementarity determining regions, and you can see they're intact within these large peptides. So we maintain the integrity of these key CDR regions. And so this was a look at a couple different antibodies and the size of peptides that would be generated using lyc. And they're illustrated here for different mass spins, going from about a mass spin of 500 daltons up to 10,000 daltons. And this is the number of peptides that would be expected in this digest. And you can see that the majority of the key CDR peptides, those that contain that antigen specificity region, are in this larger size, which is a great fit for this middle down approach and for UVPD. And so as an example, let's look what we get after we do this lyc for one of these antibodies. This is the LC trace identified or obtained using a C8 separation. And then let's say we look at this one peptide that's eluting. Now this first spectrum shows the electrospray spectrum for that particular peptide. We see a +4 and +3 charge state. And then we do HCD analysis on that peptide in the +4 charge state. The HCD spectrum is not bad. We see a lot of BY ions, but there's actually regions of the peptide which are not well sequenced by HCD. ETD, in fact, performs a little worse for this. We, again, isolated the +4 charge state for that same peptide. ETD, you see a lot of charge reduction and some fragment ions, mainly at the N terminus, but there's a large region in the middle that's not well sequenced. UVPD, we see the same thing, +4 charge state, same peptide. We see a more comprehensive sequence map, many more fragment ions in the ABC XYZ category. As you can see, some of these fragment ions are relatively small abundance, but they're well resolved and they have adequate signal noise to allow them to be assigned. And so we just look at a whole series of peptides from this antibody ranging in length from 2 to 4.6 kilodaltons. On this histogram, I've listed all the different peptides that were identified for each of the three complementary techniques, HCD, ETD, and UVPD. And we've plotted them as bars. The HCD and ETD are on the lighter color green. They don't show up so well in this room, but the dark bars are the UVPD results. And they're plotted as a function of E-values, which is a confidence type score generated using a ProSite top-down search algorithm. And you can see that the higher the score represents a greater level of confidence. So in this histogram, you're looking for higher bars. And as I said, the peptides go from increasing length from left to right. And you can see in almost across the board, the UVPD bars are the higher bars, in part because we're generating this larger sequence or series of fragment ions, ABC, XYZ, that really allow greater confidence in assigning the sequences. And so one example here, another antigen binding peptide from another antibody. This is the small peptide that elutes here. This is the electrospray mass spectrum obtained at that elution point. And then when we isolate the +6 charge state, we have a very nice UV photodissociation fragmentation pattern. This peptide actually is 8,796 Dalton, so it's one of the larger middle down size peptides. And we see very good sequence coverage throughout that peptide sequence, which allows us to identify the peptide, and this complementarity region goes from this valine all the way up to the second valine. It's completely intact. And so just putting all these middle down size peptides together, for this particular antibody, we were able to obtain 95% coverage of the light chain and 96% coverage of the heavy chain using this UV photodissociation method. So I think my time's almost up, so I'll say just a quick reminders to summarize the work. The UVPD really does offer a very fast, efficient dissociation of both positive and negative ions. The great diversity of fragmentation helps, in many cases, give high confidence in identifying and characterizing proteins. And it certainly seems to be a very promising activation method for the whole gamut, from bottom up to top down proteomics. And finally, to acknowledge the group members who actually do the heavy lifting in the lab. I've got a great group, and we're, I'm really delighted to work with them. We also have a number of collaborators, and that's really key for this work because it does provide us with interesting biological questions to try to resolve with our methods. And finally, the funding agencies who keep this work supported. And so finally, I'm happy to answer questions. We always try to do the question was, when we compare different techniques such as the high energy collision, the CID or the ETD, do we optimize? And yes, we separately optimize just to try to get the best results for each. Because not only are we trying to do a fair comparison, but we're also trying to use complementary methods to really extend the amount of data by combining methods. So we want the best out of each. Well, for the top-down work, we've been collaborating with Neil Kelleher's group to adapt ProCyte to extend the types of ions that can be identified. So that's been our main go-to method for the top-down. For the bottom-up methods, we've collaborated with a person named Wazoo who developed Mass Matrix. And that's, he was gracious enough to reprogram it and optimize it for negative mode, which is not something you commonly find with algorithms. So that's Mass Matrix. Yeah, the question was, I think Vicky's asking about perhaps secondary fragmentation for fragment ions that might happen to be formed and then absorb another photon and fragment further. And that's certainly very possible. I would say, especially in the top-down methods, lately we've used just a single pulse, single 5 nanosecond pulse to, in fact, minimize some of that secondary fragmentation. Because if you do have multiple pulses, 2, 3, 5 pulses, you can obliterate your fragment ions into smaller and smaller pieces until they're not meaningful. That it seems to be, we actually use more pulses for our peptides than we do for our proteins. So it's definitely some charge dependence there too. Thus far, we've only had a limited sets of antibodies available to do some of these studies, and we've stuck with fairly pure monoclonal ones to, to streamline what we're, and test our methods. But we do have a collaborator who would very much be interested in looking at the whole complement of serum antibodies, and that will be a huge challenge that I'm not sure we're quite ready yet, but it's waiting out there for us. Yes, the question is using UVPD for phosphorylated proteins like intact proteins. Yes, we have, and I think although it's maybe difficult to rationalize, in fact, using UVPD with very high energy photons, it's such a fast activation process, throwing many ions into electronic excited states, we don't see necessarily loss of phosphorylation as the first thing. Many of the fragments formed retain the modifications. So it's, many people would have thought maybe the post-translational modifications would be the first thing to be kicked out. That's not necessarily true when you're using some of these excited state type methods.
We're for break now, so let's thank Jenny.